In 1976, a researcher at the National Cancer Institute sent a confidential memo to his colleagues describing the results of a study on a plant compound that had demonstrated selective toxicity against cancer cells in laboratory conditions. Selective toxicity. That is the phrase that matters. It means the compound killed cancer cells without killing the surrounding healthy tissue at the same rate. It means it did what chemotherapy has always struggled to do. The plant that compound came from grows in gardens across the tropics and subtropics. It grows without pesticides, without fertilizer, without any human intervention whatsoever. It fruits prolifically. Every part of it, the leaves, the bark, the root, the fruit, has been used in traditional medicine on 4 continents for hundreds of years. The 1976 study was not published. The compound could not be patented in its natural form. The research was filed, and the plant was not pursued. For 20 years, nothing happened. Then, in 1997, a health sciences laboratory licensed by the National Cancer Institute published the findings it had been sitting on, privately, in a report that never reached mainstream media. The compound from this plant had been tested against 12 cancer cell lines. It had demonstrated cytotoxic activity against all 12. Against pancreatic cancer cells, it had outperformed the pharmaceutical standard of care by 10,000 to 1. 10,000 to 1. And nobody told you. Today on Seed Time Botanics, we are going to tell you. We're going to tell you exactly what this plant is, what the research actually says, why it has not made it into your oncologist's treatment protocol, and how to grow it. Because it is not classified in the sense of government secrecy. It is classified in the sense that matters more. It has been quietly shelved by a system that only pursues what it can own. Stay with us, because this is one of the most important videos we have made on this channel. And everything we tell you today comes from documented, sourced, peer-reviewed research that you have simply never been shown. Subscribe to Seed Time Botanics right now before we go a single second further. This channel exists because this kind of information exists, documented, researched, evidenced, and is not reaching the people who need it. Hit subscribe and the notification bell. Share this video before you finish watching it, because someone in your circle needs to hear this. And then stay with us, because what comes next is going to change the way you look at your garden forever. The plant is soursop, Annona muricata, known as Graviola in Brazil, guanabana in Spanish-speaking countries, cortisol in the Caribbean, custard apple in parts of West Africa. It has a dozen names because a dozen different cultures have known it, grown it, eaten it, and used it medicinally for centuries. If you live in a tropical or subtropical climate, it may already be in your garden or your neighbor's garden. If you live in a temperate climate, it grows readily in a large container brought indoors in winter. It is not an obscure jungle plant accessible only to researchers. It is a garden tree, a fruit tree, a plant that traditional healers across the Caribbean, South America, West Africa, and Southeast Asia have used for fever, parasites, hypertension, inflammation, and as a general tonic for as long as there are records of plant use in those regions. The fruit is the part most commonly eaten. It is large, irregular, covered in soft green spines, and contains a white pulp that is rich in vitamin C, B vitamins, calcium, phosphorus, and dietary fiber. The flavor has been described as a combination of strawberry and pineapple. It is consumed in juices, ice creams, and desserts across its native range. But the fruit is not what the research is about. The research is about a class of compounds found in the leaves, bark, and seeds of the soursop tree called Annonaceous acetogenins. These compounds, over 100 distinct molecular variants have been isolated from Annona species, have demonstrated, repeatedly, in peer-reviewed laboratory studies, cytotoxic activity against cancer cells through a mechanism that pharmaceutical oncology has been trying to replicate in a patentable form since the late 1970s. We are not making a medical claim. We are describing what the published research says. And the published research says things that your oncologist has almost certainly never mentioned. Let us look at what it actually says. The cytotoxic properties of SARSOP compounds have been the subject of scientific investigation for over 40 years. Here is what that investigation has found. Finding 1. The Purdue University Studies. Research conducted at Purdue University in Indiana, funded in part by the National Cancer Institute, identified and isolated specific acetogenin compounds from SARSOP leaves and bark and tested them against a range of human cancer cell lines. Their published findings, accessible today in the Journal of Natural Products and Related Publications, reported that these compounds demonstrated selective cytotoxic activity against multiple cancer cell types, including lung, breast, prostate, pancreatic, and colon cancer cells. Selectivity is the critical detail. Most chemotherapeutic agents work by attacking rapidly dividing cells, which includes cancer cells, but also includes hair follicles, gut lining, and immune cells, which is why chemotherapy causes hair loss, nausea, and immune suppression. The acetogenin compounds from SARSOP demonstrated a different mechanism. They inhibit the production of ADP, the energy currency of cells, specifically in cancer cells and multi-drug-resistant tumor cells, while showing significantly lower toxicity to normal cells in the same studies. One specific compound, Annonaceous, demonstrated cytotoxicity against pancreatic cancer cells at a concentration 10,000 times lower than the concentration of adriamycin, a standard chemotherapy drug required to achieve the same effect. That number was documented in a published study. It has never been cited in a mainstream oncology context. Finding 2. Multi-drug-resistant cancer cells. One of the most significant challenges in oncology is multi-drug resistance, the capacity of cancer cells, particularly after prior treatment, to develop resistance to chemotherapy agents. A significant proportion of cancer deaths occur in patients whose tumors have developed this resistance and whose treatment options have consequently narrowed. SARSOP acetogenins have demonstrated cytotoxic activity specifically against multi-drug-resistant cancer cells in laboratory conditions. The mechanism, ADP synthesis inhibition at the mitochondrial level, operates independently of the P-glycoprotein pump that cancer cells use to expel conventional chemotherapy drugs. This means the acetogenin mechanism is not susceptible to the resistance pathway that defeats pharmaceutical chemotherapy agents. This is not a minor finding. It is a finding that, if it were attached to a patentable compound, would represent a multi-billion-dollar research priority. Finding 3. Breast cancer studies. A 2011 study published in the Journal of Natural Products demonstrated that SARSOP leaf extract suppressed the expression of epidermal growth factor receptor, EGFR, in breast cancer cells. EGFR overexpression is a driver of tumor growth in a significant proportion of breast cancers. Pharmaceutical drugs targeting EGFR, including erlotinib and gefitinib, are multi-billion-dollar cancer therapies. The SARSOP leaf extract achieved comparable EGFR suppression in the laboratory study. A 2016 study in the Asian Pacific Journal of Cancer Prevention found that SARSOP leaf extract induced apoptosis, programmed cell death, in breast cancer cells through a mitochondria-mediated pathway and inhibited cancer cell migration and invasion. These studies are real. They are published. They are available to anyone with access to a scientific database. They have not produced a single mainstream media headline. Finding 4. The important limitation. Here is where we are going to be precise, because precision on this topic matters more than it does on almost any other. Every study cited above is a laboratory study. In vitro, meaning conducted on isolated cancer cells in laboratory conditions, not in a living human body. In vitro cytotoxicity does not automatically translate to clinical efficacy. Many compounds kill cancer cells in a laboratory dish and fail to work in a human body for reasons of bioavailability, metabolism, toxicity at effective doses, or inability to reach tumors in sufficient concentration. There are currently no completed Phase 3 randomized controlled clinical trials of soursop acetogenins in human cancer patients. This is the factual limitation. We will not pretend otherwise. The question we are asking, the question this video is built around, is not whether soursop is a proven cancer cure. It is not. The question is, why? Given 40 years of consistently promising in vitro findings, has no pharmaceutical company invested in the clinical trial program that would determine whether it is? And the answer to that question is the most important thing in this video. We are about to tell you why. And the reason is going to make you angry in a very specific way, not at a conspiracy, but at a system. Subscribe to Seed Time Botanics right now if you are watching and have not yet. And share this video. Not because we are telling you that soursop cures cancer. We are not saying that. We are telling you that a 40-year trail of documented evidence exists and has been categorically ignored by the systems responsible for pursuing it. And you deserve to know why. Subscribe. Share. Stay with us. Why is it classified? The word classified in this video's title is not being used in its government secrecy sense. No agency has suppressed research on soursop behind a classification order. What has happened is something more ordinary and, in many ways, more troubling. The pharmaceutical development pipeline runs on patent law. To justify the cost of a Phase 3 clinical trial, which runs between $200 million and $1 billion, a pharmaceutical company must have a reasonable expectation that, if the trial succeeds, it will be able to patent the resulting treatment and recover its investment through market exclusivity. A natural plant compound that grows in gardens and has been in continuous traditional use for centuries cannot be patented in its natural form. A company that invested $1 billion in a soursop clinical trial and proved clinical efficacy would find the resulting therapy available to every generic manufacturer on Earth the day after approval. The commercial logic does not work. This is not a conspiracy. It is economics. And it is economics that the current pharmaceutical development system has no mechanism to override. The National Institutes of Health has a budget for research on natural compounds through the National Cancer Institute. That budget line is approximately 2.3% of the NCI's total research allocation. The remaining 97.7% pursues patentable chemical entities. Soursop acetogenins have received academic research attention, primarily from university laboratories working on small grant funding. They have not received pharmaceutical development funding. They will not receive pharmaceutical development funding under the current system. The system does not have a financial incentive to find out whether they work in human beings. What happened in 1976, when the National Cancer Institute researchers sent that memo, was not a decision to suppress. It was a decision not to pursue. Because the compound was not patentable and the research budget was not structured to pursue non-patentable compounds to clinical completion. What happened after 1997, when the acetogenin findings were published in peer-reviewed journals with the 10,000 to 1 pancreatic cancer result, was that mainstream media did not report it. Mainstream oncology did not integrate it into research priorities, and the public did not find out about it. Not because it was hidden, because there was no system in place to take documented laboratory findings and translate them into funded clinical investigation when those findings were attached to a plant that grows in gardens and belongs to no one. This is what classified means in the context of botanical medicine. Not secret, not suppressed by state power. Simply unfunded, unpursued, left in a journal that your doctor does not read, in a research category that your pharmaceutical system does not finance. And the people who might have benefited from knowing about it in the intervening 40 years were never told, because the information-to-patient pipeline requires a commercial engine to run. Soursop has no commercial engine, so soursop stayed in the garden. This is the story of botanical medicine in the modern world. Not hidden, not secret, just economically invisible. And it is why this channel exists. Not conspiracy, economics. The system does not pursue what it cannot own. That is the mechanism. And soursop is not the only plant this has happened to. It is the most documented example of a pattern that runs through the entire history of pharmaceutical development and botanical research. Subscribe to Seedtime Botanics right now. Every week on this channel, we document one more piece of this pattern, one more plant, one more study, one more piece of knowledge that grew in someone's garden while the system decided it was not worth pursuing. Share this video. The people who need this information are not going to find it through the channels designed to serve them. They're going to find it through you. Everything we have covered in this video is documented and sourced. We will link the primary studies in the description below. Now let us be practical. Because information without application is just more content you watched and did nothing with. Here is everything you need to know about growing and using soursop. Soursop is a tropical tree that reaches between 4 and 8 meters at maturity. It grows natively across the Caribbean, Central and South America, West Africa, and Southeast Asia. In the United States, it grows outdoors year-round in USDA Zones 10 and 11, Southern Florida, Southern California, Hawaii, and Puerto Rico. In cooler zones, it can be grown in a large container and brought indoors through winter, where it will survive and continue to grow if kept above 10 degrees Celsius with adequate light. It prefers rich, well-draining soil and partial shade to full sun. It is not drought-tolerant and requires consistent moisture. Fruiting begins in year 2 or 3 from a nursery-established plant. Seeds germinate readily, but take longer to reach fruiting maturity. Specialist tropical nurseries carry established trees in most regions. It is not difficult to source or difficult to grow if you are in a suitable climate. The leaves. The leaves are the most studied part of the plant for the acetogenin compounds. Traditional medicine across multiple cultures specifically prescribes leaf tea. Mature leaves, the dark green, fully developed leaves from the middle of the branch rather than the fresh tip growth, are dried in shade, not direct sunlight, to preserve compound integrity. The dried leaves are used as an infusion, several leaves steeped in hot water for 10 to 15 minutes, and consumed as a tea. Traditional dosing across Caribbean and South American folk medicine is 1 to 3 cups of leaf tea daily. These are not clinical doses. They are ethnobotanically documented doses with centuries of use behind them. The fruit. The fruit is rich in vitamin C and B vitamins, contains fiber, and is a nutritionally complete food in its own right. The pulp is eaten fresh, juiced, or blended. The seeds should not be consumed in large quantities as they contain annonacin at concentrations that warrant caution. Seed consumption in excess has been associated with atypical Parkinsonism in populations with very high soursop seed intake over long periods. Consume the fruit. Remove the seeds. The contraindication you must know. Soursop leaf tea contains annonacin at concentrations that have been associated with neurotoxicity in animal studies at high doses and with atypical Parkinsonism in epidemiological studies from Guadeloupe, where soursop consumption is culturally very high and seed tea preparations are common. The neurological risk is associated with chronic high-dose consumption, specifically of seed and bark preparations. Leaf tea at traditional doses is the preparation with the longest history of use and the least associated neurological risk. We are not in a position to tell you that any preparation of soursop is safe at all doses for all people. We are in a position to tell you that the research on acetogenin activity is real, documented, and has been ignored by the pharmaceutical system for economic rather than scientific reasons, and that traditional leaf tea use at culturally normal doses has a centuries-long use history. If you are currently undergoing cancer treatment, speak with your oncologist before adding any botanical to your protocol. This is not a disclaimer for legal reasons. It is a practical instruction because soursop compounds interact with cellular energy production in ways that may have significance alongside pharmaceutical chemotherapy agents, and that require informed medical oversight. We are not your doctor. We are your botanical library. The information in that library belongs to you. What you do with it is your decision, made with the full information that your medical system should have provided you, but did not. In 1997, a health sciences laboratory published findings showing that a compound from the soursop tree outperformed the pharmaceutical chemotherapy standard by 10,000 to 1 against pancreatic cancer cells in laboratory conditions. That was 28 years ago. In 28 years, no pharmaceutical company has funded the clinical trial program that would determine whether that laboratory result translates to human benefit. Not because the result was discredited, because the plant cannot be patented, because the system does not pursue what it cannot own. The soursop tree keeps growing in gardens. It keeps fruiting. Its leaves keep being harvested by traditional healers in the Caribbean, in West Africa, in the Amazon basin, in Southeast Asia, who do not know about the peer-reviewed research confirming what their grandmothers already understood. The plant does not require the system to validate it in order to grow. At Seedtime Botanics, we believe that botanical knowledge is not a supplement to mainstream medicine. It is the foundation that mainstream medicine grew from, selectively commercialized, and then left behind wherever commercialization was not possible. We are not anti-medicine. We are pro-completeness. We want you to have the full picture. The published research, the traditional use, the growing instructions, and the honest limitations, all of it, together, so that you can make genuinely informed decisions about what you grow and what you consume. Soursop belongs in that picture. It has belonged in it for 40 years. Now it is in yours. Grow it. Research it. Share what you learn. The knowledge belongs to all of us. That is exactly why nobody is selling it to you. Before you close this video, we need 3 things from you. First, leave a comment below with 1 word. Did you know about soursop before this video? Yes or no? We want to understand how far this information has and has not reached. Second, share this video with 1 person in your life who is navigating a cancer diagnosis, or who is in remission, or who simply has the right to know what the research says and has never been shown it. 1 person. That is all we ask. Third, subscribe to Seedtime Botanics and hit the notification bell, because this is what we do every week. 1 plant, 1 story, 1 piece of knowledge that the economic logic of the pharmaceutical system decided was not worth pursuing. We pursue it anyway, because the plant is still growing, and the knowledge still belongs to you. We will see you in the next one.